and we will cover the topic of biochemistry. So let's get started with the different elements that are found in the human body. So beginning with the first element that is written over here, which is carbon. Now, in our body, about 19% of our body is made up of carbon. And carbon is a very important part of all organic compounds. Carbohydrates, proteins, and lots of other organic compounds as well. In fact, even DNA and RNA. About 10% of our body is made up of hydrogen, which is an important part of organic compounds and also of water. About 65% of our body is made up of oxygen, which is again an important part of organic compounds, especially sugars, and of water, and it is also used in aerobic respiration so that our cells can use the oxygen to form energy. Coming to nitrogen, about 3% of the body is made up of nitrogen, which is an important part of amino acids. And amino acids are an important component of proteins. The next element is sulfur, which is present in trace amounts, and it is found in some kind of amino acids, not in all of them. So let's do two examples of those. One is cysteine, and the other is methionine. So in these two amino acids, sulfur is present. The next important element is calcium, which is present in the bones and in the teeth, and it also helps in generating or carrying the nerve impulses from one neuron to the next one. Then coming to the next one, which is potassium and sodium over here. So the last ones, these are used as pumps, and they also help in generating the nerve impulses. So they're helpful for the neurons to function. Then coming back to phosphorus, an important part of the nucleic acids, both DNA and RNA, and also an important part of ATP, which is adenosine triphosphate, the component through which energy is released in our body. And the one that's left over is iron, which is an important part of hemoglobin, and so it is important for the transport of oxygen in our body. So that is the information that you need to know about the different elements. And since SAT is an objective test, you do not need to concern yourself with how to explain the different, let's say, explanations or how processes are happening. So you need to know more objective information as compared to the subjective one. So let's go ahead and see what are the different kinds of bonding that are available in different organic compounds. So these are the 6 main kinds of bonds that are found in the human body: covalent bonds, hydrogen bonds, peptide bonds, phosphodiester bonds, ester linkage, and glycosidic linkage. Let's start with covalent bonds. Now these are present between atoms of the same molecule. And they can be single, they can be double, they can be triple as well. An example of this is the water molecule, where oxygen is covalently bonded to 2 hydrogen atoms, and this covalent bond exists through sharing of electrons. But in some molecules, for example water, the sharing is not equal, so as a result, the electrons are pulled towards oxygen and away from hydrogen. So oxygen develops a slight negative charge, and hydrogen, they develop a slight positive charge. So it's not one complete charge, but just a slight positive charge. So covalent bonds can exist between polar molecules. For example, water, where you have one negative side and the other is a positive side. And these covalent bonds can also exist between non-polar molecules, for example, the hydrogen gas. There is no polarity between them, so it's a simple non-polar covalent bond. The second important bond is the hydrogen bond. This is specifically mentioned and present in the DNA double helix. So in the ladder-like structure of the DNA, between the different base pairs, you have the hydrogen bonding. So we can say the hydrogen bond is the one that holds the two strands of DNA together, and also between different molecules of water. For example, this is one molecule, and this is another molecule, and this is another molecule. Between the hydrogen of one molecule and the oxygen of the other molecule, this is also another location where hydrogen bonds are found. The next important bond is the peptide bond, which is found between amino acids in a polypeptide chain. So a polypeptide chain or a protein basically consists of a lot of amino acids joined together through the peptide bonds. The next bond is the phosphodiester bond, which is present between different nucleotides of the DNA. So let me draw a rough structure for that. Let's suppose this is the phosphate molecule, and this is the sugar molecule, and this is the nitrogenous base. Now this bond over here that connects the sugar molecule to the next nucleotide's phosphate is going to be the phosphodiester bond. The next one is ester linkage, which is present between fatty acids and glycerol when they combine via the condensation reactions to give rise to a triglyceride. So we'll be doing these reactions in a little while from now, but right now you just need to know what is an ester linkage, which is present in case of lipids, that is triglycerides. Glycosidic linkage, the last kind of bond that we have to do here, is present between the smaller sugar molecules that are part of a bigger sugar molecule. For example, glucose will be joined to glucose via a glycosidic linkage. So that is all that you need to know about the different bonds. The next important thing that you need to know is the difference between two important terms. One is hydrophobic and one is hydrophilic. So as the name suggests, hydrophobic molecules are those who hate water, or basically they are repelled by water. So examples of these are lipids, oils, and waxes. So basically compounds that do not dissolve in water. On the other hand, hydrophilic are those who love water. And examples of hydrophilic compounds include starch, which is a carbohydrate. So that is what you need to know about hydrophobic and hydrophilic. And we'll be using these concepts in a lot of other related topics as well. But the next important topic that we have to cover in this video is the different characteristics of water. So these are the important characteristics of water, or its different properties that you need to be aware of. High specific heat. This refers to the amount of heat that must be absorbed in order for 1 gram of water to increase its temperature by 1 degree Celsius. So that is what high specific heat means. That means that water takes a lot of heat before it can get, before its temperature can increase by 1 degree. And similarly, it needs to let out a lot of heat before its temperature reduces. That means it is not easy, it's not really quick to heat or cool down water. It takes a lot of time. The next important property is the high heat of vaporization. So that refers to the amount of heat that must be absorbed. So the amount of heat absorbed to convert 1 gram of water from liquid to gas at room temperature. So that is the high heat of vaporization, which means that water does not boil very easily, it does not vaporize very easily. Then high heat of fusion, which is the amount of heat that needs to be lost to convert 1 gram of water from liquid to solid, which is basically ice, at the same temperature. So that means water does not get frozen into ice really quickly. So that helps us human beings in cold temperatures because our blood does not become frozen very soon. Then high adhesion properties, that means water, it sticks to other molecules very easily. So water sticks to other molecules, specifically polar molecules, very easily. And cohesive properties mean that water sticks to water molecules very easily. So water sticks to water molecules very easily, which is useful in transpiration. Then is universal solvent. Because it is highly polar, water can dissolve a number of polar and ionic substances. Because ions will also be charged, so that means they will be either positively charged or negatively charged, which will be attracted by the poles in the water molecule. The next important property, it becomes less dense on being frozen, which is the reason why ice floats on the water. Because when water becomes frozen into ice, it becomes less dense and the ice floats. And this is very good for the aquatic life or life in areas like Antarctica and the Arctic Circle. So it helps to prevent the oceans from freezing. And water is transparent, which is really good for the aquatic plants as sunlight can reach the aquatic plants. So sunlight reaches aquatic plants, and that is a benefit of water being transparent. So those are the properties of water. The next important thing you need to know in this video is what is pH. So the word pH is actually a measure of the acidity or the alkalinity of any substance. So the lesser the pH value, the more acidic a substance is. The higher a pH value, the more alkaline the substance is. So let's see a few important substances that are found in general life, whether they are acidic or alkaline. Let's start with the one that is in the middle, which is going to be pure water. So that falls under the category of 7. So 7 stands for neutral, and an example of that is the water that we drink. Now starting from the acidic one, the different molecules that are present or the different substances that are present on the acidic range include, first of all, battery acid, which is highly acidic, and then gastric acid, which is secreted in our stomachs, is also really acidic, although not as acidic as battery acid. And then hydrochloric acid, so let me just represent it by HCl. Then the compound or the substance with 4 acidity is soda. For example, any aerated soft drink. The one with 5 includes acid rain. And the substance with an acidity of 6 is black coffee. And between 6 and 7 is another compound, which is urine or saliva. So the human urine and the human saliva have an acidity, and not very acidic, but they're not even neutral. And they have a low pH, but not very low either. It's between 6 and 7. So coming to the other side of the pH scale, which represents the alkaline substances, let's start from this. So seawater would occupy this position. It's a little alkaline. Next, it's going to be baking soda, which is alkaline. So baking soda. And the next thing is milk of magnesium, which comes between 10 and 11. So I've missed 11 over here. Let me write that down, which is milk of magnesium. And between 11 and 12 lies ammonia, which is NH3, a pungent gas. Between 12 to 13 is soapy water, closer to 12. And 13 would be bleach. And finally, the most alkaline is a drain cleaner. So this will give you a general idea that the neutral substances are least harmful for us, and the ones that are highly acidic or highly alkaline are detrimental for our health. So that is about what the pH scale tells us. The next important thing that we need to cover in this video is the organic compounds, and we're going to cover carbohydrates next. So carbohydrates are organic compounds that are responsible for short-term energy storage. So not in terms of days or weeks or months, but in terms of hours and minutes. So they provide short-term energy. In case of plants, they are stored in the form of starch. But in case of animals, they are produced in the form of glucose and stored in the form of glycogen in the liver. And as and when we need glucose, the glycogen keeps getting converted to glucose. And that is a process that we will actually study when we do the endocrine system, how the two different hormones secreted by the liver cells and the pancreas cells, they help in regulating the amounts of glucose and glycogen. But right now you need to know that it helps in short-term energy storage, and the amount of energy that they give is close to 4 calories per gram. So 1 gram of carbohydrates gives you 4 calories of energy, and they are of 3 types. The ones that are simple, for example this diagram over here, it consists of one ring. So these ones are known as monosaccharides. There are others that are composed of 2 rings. They will be known as disaccharides. And the ones that are composed of more than 2 rings are known as polysaccharides. Now the formula for carbohydrates, the basic structural formula is this, CH2O N. So it could be, let's suppose N could be 5. In that case we get ribose sugar, which is C5H10O5. Or N can be 6, in which case we get glucose, C6H12O6. Now different molecules, the different monosaccharides, they combine and form disaccharides. And this reaction is known as a condensation reaction. So basically 2 small molecules or 2 rings are going to form one big molecule and going to release 1 molecule of water. So this is known as a condensation reaction. And the opposite of a condensation is a hydrolysis, where in the presence of water, a disaccharide or a bigger unit will break down into 2 component units. Let's study a few examples of this process of condensation. For example, glucose and glucose give rise to maltose plus water. Similarly, glucose plus galactose give rise to lactose plus water. Another example, glucose plus fructose combine and give rise to sucrose plus water. So if the reactions go from the smaller components to the bigger ones, it is known as condensation. But when it goes from these bigger compounds to the smaller ones, it is known as hydrolysis. So let's see how this reaction actually takes place. So what happens in the condensation reaction is shown by this diagram. So you have 2 molecules of this compound, which is sugar, and basically this OH group of one sugar molecule and the next molecule's OH group, they combine and take part in the condensation reaction. So you have OH and you have OH over here. This gives rise to the water molecule that you get, and the remaining oxygen is shared common between them through this covalent bond that you can see over here, and specifically the name that we give to this is the glycosidic linkage. So this is how a glycosidic linkage is made, and you get a bigger molecule which is maltose. So let's do a few examples of carbohydrates. So an example of monosaccharides is glucose, which is used for short-term energy in animals, and fructose, which is fruit sugar found in honey and onions, and it's a good source of energy. Example of disaccharides would include lactose, which is milk sugar used in animals, and sucrose, which is the transportable form of sugar used in plants. And examples of polysaccharides include chitin, from which the exoskeleton of insects is made, and glycogen, which is a form in which glucose is stored in our bodies, and cellulose, which is a component of the cell wall of plants, and even starch, the form in which carbohydrates are stored in plants. So that is all that you need to know about carbohydrates. The next important compound is lipids. Now lipids include the compounds known as fats, oils, and waxes. Now there are two kinds of lipids, saturated and unsaturated. Let's study what is the difference between saturated and unsaturated lipids. The saturated ones are derived from animals. So animal fat is saturated and it is solid at room temperature. For example, the fat inside our body is all saturated. They don't have any double bonds between the chains of carbon, and they're linked to heart disease. An example of this is butter. Unsaturated are derived from plants. They are liquid at room temperature and they have at least one double bond. Okay, so they have at least one double bond. An example of that would be, take any cooking oil, for example, olive oil. That will be an example of an unsaturated lipid. Now, the function of lipids is to give long-term energy storage. And unlike carbohydrates, which give 4 units of energy per gram, they give close to 9 units of energy per gram. Even they undergo the condensation and hydrolysis reactions, and the new bonds that are formed are known as ester linkages. So, this reaction of condensation, it occurs between glycerol and 3 fatty acids. And this reaction gives rise to a triglyceride and 3 molecules of water. So let's see an example of how this happens. So we have a molecule of glycerol that has 3 OH groups, and we have 3 fatty acids that also have the OH groups. And these are the chains of the fatty acids. So the condensation reaction will occur between these OH groups, and the new bonds will be formed with oxygen, which is commonly shared between the two groups. So let's see how the molecule will look like. So this is what the resulting molecule will look like. So hydrogen and oxygen will come out and will form the 3 water molecules, and the remaining oxygen will be shared with these 2 different molecules, and this will give rise to a triglyceride. So that is what happens in a condensation reaction for lipids. The next important compound that we have to do, the next important organic compound, is proteins. So this is what a structural unit, which is the amino acid, it looks like. And this part, the NH2, this one is known as the amino group. So let me put it down over here. This is the amino group. This part is known as the carboxyl group. The R over here is the variable side chain, which varies from one amino acid to the other one. The remaining part remains constant. So proteins are the basic structural units of any organism, and the basic unit of a protein is an amino acid. And there are a few structures you need to be aware of. This is the first level structure of a protein. So these little round things that you can see are basically amino acids. So all amino acids are linked together in a chain. This is known as a primary structure of a protein, or the 1-degree structure. So this is nothing but a sequence or a chain of amino acids. The second structure that you can see is when this chain gets arranged into the form of a helix. We call this an alpha helix, and this is the next level structure, so we call it the secondary protein structure. Or this is a 2-degree structure. It occurs when the sequence of amino acids are linked by hydrogen bonds between them. The next structure is this that you can see. This is known as a tertiary protein structure. So this is basically made, now this was the alpha helix. The alpha helix is going to be situated like this. So throughout this tertiary structure, this is the alpha helix that I'm drawing right now. So the alpha helix itself is presented in a convoluted way, which is the tertiary level or the 3-degree level, and this is when the certain attractions are present between the alpha helixes. And the next level is going to be a quaternary protein structure, which is there in case the protein has more than one chain, so we call it a quaternary structure. So similar to this structure, you will have one more structure that will be going side by side with this one. So you'll be basically having two structures, and let me just draw that a little bit. So this structure that you can see now, you can see there are two groups, so two different chains of the protein. You can see this. So this is the quaternary structure, which is 4-degree structure, and it's seen only in some proteins, for example, in case of human beings. And in case of the special protein known as hemoglobin. So the reaction that takes place within these smaller amino acids that gives rise to these polypeptide chains is also known as a condensation reaction, and the bond that is formed is the peptide bond. So basically the reaction is between the carboxyl group of one amino acid and the hydrogen of the amino group of the next one. So let me draw that in short. So this new compound that you can see, the new molecule, so this was one amino acid, this was the other one. Between the carboxyl group of this amino acid and the amino group of this amino acid, the water molecule has been taken out, and the carbon and the nitrogen instead form the bond. And this bond over here is known as the peptide bond. So that is all that you need to know in case of proteins. Let's move on to the next molecule, which is a kind of protein known as an enzyme. Enzymes are basically large proteins that speed up the rate of reactions by lowering the activation energy. That means the reactions, they get activated really quickly. They are helped by cofactors, which are minerals, and also by coenzymes, which are vitamins. Now, to explain enzymes, there were two models that were introduced. Initially, the model that was brought forward was the lock and key model, but it became outdated, and the new model was induced fit model. So let's see what happens in each one of them. So this is what happens in a lock and key model. The enzyme is there, and the two different reactants are there. These reactants are going to enter these parts of the enzyme, and this complex is known as the enzyme reactant complex. Now the reactants will come here and get involved in a reaction that will give rise to enzyme product complex, and then the final product will be released outside the enzyme. So as you can see that there is no difference that can be seen in the shape of the enzyme. But why this model got outdated was because in the real life, the enzyme does undergo some structural change. So that is why it's called an induced fit. It's not a natural fit. The shape of the enzyme changes a little bit to accommodate the different reactants. Let's see how that happens. So this is what happens in induced fit. As you can see, the shape of the enzyme is not really compatible with the reactants right now. They are moving over here. But once they come close, the enzyme's shape, it changes. It becomes the enzyme reactant or the enzyme substrate complex. Then it gets converted into the enzyme product complex. And when the product leaves the enzyme comes back to its wavy shape. So that is the induced fit model. Now, there are a number of different factors that affect the activity of an enzyme, and we need to go through them. For example, temperature. Now, up to a certain limit, the activity of the enzyme, which I'm representing here by A, will increase as the temperature is increased. But after a point of time, when temperature becomes too much, it's going to drop because the enzyme gets denatured. Denatured means a physical change will happen in its physical properties, a permanent change, and the enzyme would not be active as it was earlier. So the enzymatic activity, which is the rate of reaction, it increases with the increasing temperature until the enzyme is denatured by heat and it gets inactivated. And at this point, the reaction falls steeply. So if the enzyme was, let's suppose this was the structure of the enzyme earlier, after denaturation, it becomes like this. So it's not useful anymore. The next important factor is pH. Now for each enzyme, the activity depends on the pH, and for each enzyme, there's a different pH. For example, an enzyme that works best in acidic pH would slowly and slowly increase when that pH level comes, and when it goes, it would decrease. So the graph is more or less like a proper curve. It does not fall steeply. So there is a specific point where the reaction takes place at the maximum rate. Then the third important factor is substrate concentration. So up to a certain level, the activity will increase as the substrate concentration is increased. But after that, the rate of reaction will increase only till the active sites on all enzyme molecules are filled, after which the maximum rate of reaction is reached and the enzyme activity will continue like this, so it will become a plateau. So that is going to happen in case of substrate concentration. One more thing that is left in this video to be covered is the organic compound known as a prion. So it's a very small concept. Prions are basically proteins that cause disease. They can be present in bacteria or they can be present in viruses, but these are proteins that cause a disease, and usually they are a misfolded version of a normal protein. So that is what the prions are. And that is all that we will be covering in this video. The next lesson that you need to watch after this is known as the cell, and we'll be discussing different organelles that are found in the cell.